Now is the point where we start getting into what you thought you were getting into when you signed up for biology. You're going to learn about the cell. Finally, I know this has taken forever to get here. So, I'm going to give you some brief background information about it. You don't need to write most of this down because it's history. And I'm not going to really quiz you on the history aspect as much as I want you to see the bigger pictures. So I'll tell you when it's really important to start writing things down again. So here's the deal. 1665. A new tool of science was created called the microscope. We've heard of these things, right? We have a whole bunch of them back there. It was created by a gentleman named Robert Hooke, and like any good scientist, so do you guys know how a light microscope works, basically? You've got one lens stacked on top of another lens. You look down through one, and it magnifies through the other as well. Have you ever looked through somebody else's glasses before? It's kind of weird. Thank you. It's kind of weird. You start to see like things that are far away, and they look close up, and they look all distorted. People have had this magnification technology for a long time. And the Chinese were using it for probably about 2,000 years. You know what a spyglass is? Yeah. If not, think of what pirates use when they're like looking at other ships like that.
Or the movie Ants, where there's a bug's eye.
It was one of those. So you're looking through the spyglass. One end has a curved lens. The other end has a curved lens. And it magnifies both. So you see things up really close. So what he was using was sort of piggybacking on that technology. Because have you guys ever played with a spyglass before? If you do it, you do two things. One, you see things that are really far away. And then immediately you like look down. You're like, what does it look like up close? It's pretty much what Hooke did. He's like, all right, I got these two lenses, and now I'm looking down. And what did he have nearby? A cork off a wine bottle. Now, I'm not going to make any disparaging remarks about Hooke, but, you know, having that wine bottle sitting in his lab's kind of cool. It's like, I'm looking around with this, looking at this. Oh, hey, look at that. And what he noticed in the cork were these, what looked like chambers. They look like squares. And he said, those look like, as you would at the time, the cells of a monastery. And thus he called them cells. It's where monks would live. All right, about 100 years later, we had this microscope now. It didn't have much of a purpose. I mean, really, you couldn't do much because the glass in it was so poor. If you've ever looked through the bottom of a bottle, any kind of bottle, I'm not, again, college students, I understand, but if you're looking through the bottom of a bottle trying to find that last drop of whatever, you'll notice it's very distorted. You can't really see other people. They're kind of all blurred and weird.
Well, optical glass wasn't invented until like the 1800s.
Yeah, 1775, actually.
Almost,
Yeah, exactly when. Exactly.
Almost 1800s.
Right. So what they ended up doing about 100 years later was coming up with a new glass-making technology where they're coming up with this optical lenses that can do more. Gentleman by the name of Anton von Leeuwenhoek. There are several weird punctuation things that go in that name that I just didn't have the right characters on the computer to put in. So there is definitely a hawking in the back of the throat.
Sounds that don't exist in English.
Sounds that don't exist in the English language. Anton von Leeuwenhoek. He refined the techniques to make that glass. And he wanted to sell this new glassware. So what he did was, to sell it, because business, right? You give people what they want. He said, check this out. I took two lenses, I put them on top of each other. This is what's living in the water that you're drinking. And he would show people these microorganisms. It was a selling point. Scientists turned around and said, that's really cool. There's stuff living on me. You have this thing called a microbiome, in case you didn't know. The microbiome is really freaky, and if you ever get around to looking at it, you'll be amazed at how dirty people are. Not how dirty, but yeah, why you know people are putting on the ethanol. Things like how many of you guys are in a relationship right now? Wow. Oh wait. How many of you guys are in the it's complicated? Okay, there you go. The single's not going to cut it for you. Not for this particular discussion. Have you been in a relationship before? Yes. All right. When you're looking deeply into somebody's eyes, you know, that whole first, second, third date and then it's over thing, As you stare deeply into their eyes, you might glance at their eyelashes. Now, don't let them know, but there could be things crawling all over them. Oh, I know. Yeah, well, there's stuff literally cleaning all of your hair follicles. There's stuff crawling all over you. Now, usually, we don't... she's going to go check. It's like, is it me? Is it me? No, it's cool. She didn't. There's stuff crawling all over you. And now that we have this technology, we can look at it. You guys, a lot of you, most of you have phones. Obviously, right? You can zoom in really tight with the phone. And with the resolution they have now, if you were to hold that over even a single 10 times magnification and then zoom in even closer, you will be able to see the organisms crawling on you. Can you do that right? I don't have an extra lens.
I do. I just left it at home.
Oh.
Next time.
Yeah, no, sorry. Look, I will bring you an extra lens so you can do that. It's really interesting. In fact, when we next week we're going to do the microscopy lab.
Oh, sweet.
Please feel free. These don't have recorders on them, but if you want to take your phone and hold it up to the lens, you'll be able to take pictures of the individual cells we're looking at. I call them selfies. Right. But you'll be able to take pictures, see it, show your friends. Science is cool. So, so we've got the light microscope since about 1800. We were figuring out how the world was working and what all these little things crawling around in it were. 40 years after that, a guy named Matthias SCHLEIDEN. SCHLEIDEN's another German gentleman. He was a plant physiologist, so he's looking at plants using Leeuwenhoek's. Sorry, I need more spit. Leeuwenhoek's technology. SCHLEIDEN noticed that all plants, everywhere, all of them had this dark spot in them. And he's like, huh. I would do it in German, but I can't. Huh. Sorry. I've seen this dark spot in all of these plants. This is very interesting. He told his good friend, Theodor SCHWANN. Now, SCHWANN proposed this idea. He was an animal physiologist. He's like, I too have seen these dark spots in cells. Maybe this is a thing all cells have. And they're like, yeah, that's a good idea. They're probably having like bratwurst or something when they're talking. Beer and bratwurst, right? They proposed, SCHWANN said, I'm going to publish a paper on this. And SCHLEIDEN said, no, I came up with it. I'm going to publish a paper on it. And then they stopped being friends. Yeah. They raced. SCHLEIDEN got his published first. SCHWANN followed immediately afterwards. And between the two of them, they got this out into the public. There are these dark spots. 20 years after that, a Russian by the name of Rudolf Virchow was studying cancer cells. Cancer was bad even then. Cancer's been killing us for hundreds of years. It just seems like it's a lot worse now, and it might be. But it's probably because people are living to a much, much older age. And we're much more aware. We're able to spread the ideas. And it's not just... People don't die of natural causes anymore. Natural causes doesn't count. There's no such thing. Now it's just people die of something particular, usually cancer. So what Virchow noted was that normal cells make more normal cells. And cancer cells make more cancer cells. I know, go figure, but this is big news. Between the three of them, SCHLEIDEN, SCHWANN, and Virchow... Need more time? Everybody okay?
I like that you put all of the PowerPoints up.
Oh, yeah.
And just download it all.
Good. Now, the light microscope was super cool for its time, for 200 years. And in fact, it's still pretty cool. We still use it. Except there's a big sign on this that says, do not touch them, which is kind of weird for a biology class. We're going to touch them a lot. To hell with the sign. There are some other microscopes that have come into play recently, the electron microscopes. And you have two kinds, the scanning electron microscope and the transmission electron microscope. The scanning electron microscope works almost like, you know those lasers at a grocery store as you scan a barcode? They're coming out, and you'll notice that laser sort of follows along the contours of whatever you're scanning over it. That's what this scanning electron scope does. It scans over the contour of the cells, and it gives you a perfect three-dimensional image of those cells. The problem is you can only see the outside of the cell. A transmission electron microscope, on the other hand, fires electrons through a very, very thin section of cell. It takes a long time to make these slides, because you take an organism and you slice it. Oh, they don't survive the process. You surround it in paraffin or in a wax, and then you slice it with what's called a microtome, which slices things to about 1/1000th the width of a hair. And then you were to put that on top of a slide in absolute vacuum in a purely clean room.
How do you get a slipping obsidian scalpel through?
How do you do that?
Yeah.
It's really
Vacuum, like it would just think.
You'd think that, but what ends up happening is it's all computer regulated. So you're not actually slicing, and that it's
Reach your hand in there.
no, you don't. You do it all with these little robotic hands. It's kind of cool. Fires electrons through, and then you're able to see exactly what's happening. So what you're looking at here, these little individual sticking out pieces, these little hairs, are actually individual proteins, individual molecules. You can look at, that doesn't have the resolution to hit just one atom, but we're really close. In fact, actually, I think they do have it where you can arrange individual atoms now.
I think they have to view them with like charges, though. It's really hard.
Yeah, it's tricky. So the thing is, for most of your lives, unless you are going into advanced biology or chemistry, you're never going to use those. I don't even know where the closest. Probably actually VCU has one of these. I imagine they do. They are. Sometimes they take up one story. Sometimes they're 2 stories high. They have to have specialized rooms for them, specialized power sources. They're really tricky. I've used a scanning electron microscope exactly one time in my career, and it took me 6 months of training and 3 weeks to prepare the slide. And I got one picture. No, that's not the picture I took. I was taking pictures of the proteins on the outside of a fly's egg. Yay! All right, so now we can see stuff, primarily with these these light microscopes. Now we're going to talk about the types of cells that we see. And there are 2 broad categories: prokaryotic cells and eukaryotic cells. Prokaryotic cells have no compartmentalization. Eukaryotic cells have lots of compartments, lots of spaces where things happen. Prokaryotic cells are relatively simple. Things are just scattered all over the place, whereas eukaryotes have very, very complex structures. Do you guys know what I mean by compartmentalized? Have you heard that term before?
Yeah.
Yeah, everything has its proper place. I would describe it as you're compartmentalized. Everything has its proper place. How many of you guys have a set of drawers? Yeah, okay, you got a set of drawers. Do you have a sock drawer? Yeah. What, is there anything else that goes in your sock drawer? Yes. No, yes. What else goes in the sock drawer? Sure, underwear. That's what I do. I do like underwear, socks, t-shirts over here.
Yeah.
Well, you guys.
There are certain types of t-shirts that will go.
Right. You guys have a strategy to your drawers, right? Yes. Yeah. Some people don't. They just start cramming things in their drawers. That's your sister. Even better. This one may resonate with some of you. Hampers and clean versus dirty clothes. How many of you guys have a hamper, someplace you put dirty clothes? Yay! Do you put your dirty clothes in there? Yeah. Okay. You put them in. There's a pile of dirty clothes in that hamper. You do the laundry. You come out. What do you do with the pile of clean clothes? You put it back in the same hamper. Right. Do you then fold your clothes and put them away?
Yes.
Oh, now we have 2 distinct types of people. There's the type of people that have drawers that they are really religious about putting things away in. And then there are the type of people that have a dirty pile and a clean pile. And occasionally, an in-between that you sniff just to make sure. It happens. So, compartmentalized, the people that have those drawers and they put things away perfectly. Non-compartmentalized. It's dirt. It's clean, but it's all in a big pile. And you're just searching for a sock for 15 minutes. That's surprisingly true. My wife has recently decided, so my wife and I split the chores. My job is load and unload the dishwasher. And really my job is load the dishwasher the first time, and then she comes and fixes it because I obviously did it wrong. Because, in your lives, not that I'm off track, but in your lives, the dishwasher. You don't have a dishwasher? If you have a dishwasher, is it compartmentalized? Is there a specific place for bowls and plate? Yeah, right? That's what my wife says too.
You don't put bowls in the bottom.
You don't put bowls, yeah. Well, I was just like, it's a dishwasher. I put a dish in, it'll get clean.
But you gotta think about gravity and...
Look, there's a lot that goes into it, but my thought was, I'm not very compartmentalized. I'm just going to put these things everywhere. Wherever it fits, and it should get clean, because it's a dishwasher.
No, you clearly have not, I guess you've much better dishwasher.
No. What? The utensils, I guess, do need to go in that little utensil basket. The idea is there's a compartment and a place for everything. That's compartmentalized. That's like the eukaryotic cell. Other cells are non-compartmentalized. They don't have that place for everything to go. It says everything's everywhere. Children. God, when we're young, really, toys go everywhere. As you get older, though, right, well, as you get older, you're like, why don't you put your toys away in their proper place? Kids tend to be very non-compartmentalized. I've got my Legos, my trains, my Transformers, they're everywhere, and I will play with all of them equally.
But the Legos live on the rug with the roads and the streets and cars, and then Spider-Man lives on the dresser.
Well, no, that's the thing, though. Then the kids turn around and they're like, they lay it everywhere. The parents are like, put it in that box and put that box over there. Put it in that box and that box goes over there. Compartmentalization versus non-compartmentalization. Which is better? Neither. And no, because the people who have their clothes in two distinct piles are always going to have their clothes in two distinct piles because that's what works for them. It works just fine. Those that compartmentalize, it works fine for them. It's just two different ways of doing things. Now, with prokaryotes, all prokaryotes have three things in common. Thing number one they have in common. They all have a plasma membrane. This plasma membrane is made up of phospholipids. They've got this polar head and a nonpolar tail, so what are they termed? Amphipathic. This phospholipid bilayer is going to be filled with proteins that allow things in and out, or allow for signaling. So all prokaryotes have a plasma membrane. All of them also have what's called a nucleoid region, a place where the DNA hangs out. It's not bound by anything. It's just this looping DNA that's held generally in one place. But it could go anywhere. So it's a nucleoid region. And they all also have ribosomes. Ribosomes are proteins and nucleic acids combined together that that convert RNA to a polypeptide. Pretty much, these are little protein-making factories. So so all of them across the board have a plasma membrane, a nucleoid region, and ribosomes. Questions? That's just memorization. There's a lot of other stuff that they have that's sort of extra. Things that give them properties and things, but that not all of them share in common. Like pili. Pili are great. They're these little hair-like structures sticking out. These pili that stick out can be used for sort of grabbing onto things and pulling, walking along. They're also really important for sex between bacteria.
That's how they hold each other.
That's how they hold on tight. And they can transfer DNA across them. They're hollow. So it's almost like, I don't know, E.T. touching fingers, except DNA transports. It's a bad idea. Don't think about that. It's the idea that you can pass DNA directly from one bacteria to another through its pili. They got flagella, these whip-like structures that can move it. Acts like a motor. Cell walls A cell wall is there so that the bacteria doesn't explode when it's put in fresh water. You guys ever dropped a slug in a bucket of water? No. It gets softer and swells up like a balloon. Do you put salt on them? That's the opposite way of going. It's really skinny.
Versus,
Right. You're filling them up with osmosis. Water's flooding into them. They're filling. Okay. You guys seen The Walking Dead? All right. You remember like first season, maybe second season, when the walker fell down the well? Yeah, and it was all bloated. It's like that. It's like that. So you didn't get the idea of a slug dropped in a bucket of water, but a corpse in water. Oh yeah, we got that. So we got this corpse swelling up, the cell swelling up. The cell wall would prevent that. And then we have glycocalyx. Glycocalyx is this layer of slimy carbohydrates. It's almost like mucus. It protects the cell, makes it slide. Lots of different things in prokaryotes to protect them, let them move. So that's all I got on prokaryotic cells. I'm going to move into eukaryotic cells. Do you guys remember this picture? You guys have seen this picture before, and you had to label all the parts of it? I hate that so much. I am not going to make you do that. No, I was traumatized by this picture. And it was not even for the reason of like memorizing stupid things. I was traumatized because I realized when I went farther in biology, that's not what a cell looks like. And we're going to get to the you guys know my feelings on mitochondria and how, yeah. And they're giant mitochondria here. They're teeny tiny. I'm not going to have you memorize what these things are supposed to look like. What I will have you memorize and understand is what they do. Because each of these organelles, each of these tiny membrane-bound compartments are connected to the other ones in order to really build proteins and keep the cell moving. So we're going to start talking about each of these membrane-bound compartments. Before we do that, though, we need to talk about what makes them. So we've got this big system going on, encompassed by a plasma membrane, with a cytosol on the inside, this region on the inside, a nucleus at the center, and then an endomembrane system and something to produce the energy. The first component of that is the plasma membrane. This is the outside of the cell. This is a lipid bilayer, a phospholipid bilayer, that entirely surrounds the cell and makes up all of the organelles. Again, phospholipids. They've got hydrophilic heads, hydrophobic tails. If that's the case, hydrophilic heads, hydrophobic tails, what do you know is out there? Water in here. And if that water is, if there's a solution there, it's not just pure water, what would it be termed? Question is, yes, there is some water out here, there's some water up there. This is a solution. If it's a solution with water as the solvent, what's it termed? Aqueous, good. Oh, I thought somebody said aqueous, or aqueous, either one. Well, did you say, well, yeah, it is. So in this phospholipid bilayer, we also have proteins embedded in it. Check this out, and I want you to make this mental leap because it's kind of important and why we talked about all the chemistry. These are phospholipids. Polar head facing water, nonpolar tails, no water, polar head facing water. So far, so good, right? This is a protein embedded in it. What is the monomer of a protein? The amino acid. Those amino acids each have unique chemical properties to them. This is inside of this phospholipid bilayer. This is outside and this is outside. So tell me chemically about the amino acids right out here. They're hydrophilic. Thus these are, and those are, hydrophilic. That means that this part right here is anchored. It's not moving out of the membrane. It's not just falling out, because every time it would go to fall out, those hydrophobic amino acids push it right back in. It's held in place. Now, these proteins can help transport material across the cell. It's hard to get through a phospholipid bilayer. But if you have channels, big openings in it, stuff can get through. These proteins can help with cell signaling. Here are signals coming from outside of the cell. The only way it can interact with the inside of the cell is through interaction with the protein. So this signal comes in, binds to a protein. That protein will do something on the inside. It changes its shape because you've had a chemical change out here, changes the shape of the protein. Something might be released on the inside, and the process can happen. So proteins help that are embedded in the membrane help with cell signaling. These same proteins might help with cellular adhesion. Adhesion means what? Yeah, they stick to something else. Now, I'm not going to make you memorize integrins, selectins, cadherins, and the IG superfamily. What I put this up here was so you could see shapes, forms. What's this shape like? A cane, candy cane, or a hook, or a crook, or one of those shepherd's things. The crooks things. What do you do with those hooks?
You grab a sheep snap.
You grab a sheep snap and you pull. Or that's Velcro is made up of a whole bunch of little hooks. These hooks on these cells can reach, two cells can reach out and hook each other. And they're bound. They're holding on to each other because of the shape, the form. Here are the selectins. You'll notice it's got this ball at the end, this curved shape.
It's like Sailor Moon's staff.
It's like Sailor Moon's staff. It's like, I'm thinking more actually like a plunger. Okay. But, you know, Sailor Moon's staff, plunger, same thing.
I can't see the third dimension. Is it shaped like DNA? Or?
Is it shaped like DNA? Actually, no. It is just lumpy. It's yeah.
So it's like lumpy space.
Lumpy space, right. There you go. So what you have is on one cell you'll have a receptor, and on the other cell you'll have that selectin, and they bind like that. They just bind tight. Cadherins. Cadherins are pretty cool. They're almost like Batman's grappling hook. Because what will happen is it will pierce the other cell, and then once it pierces, open up so that they're held together tight. Because, yeah, there's a little hook on the end. And then the IG superfamily, I always think of it like an octopus's tentacle. Because you see these disulfide bonds there? Right. Well, what will happen is when it hits the other cell, those disulfide bonds break there, and they attach to the other cell. So it's sort of like they're giving each other a big hug. Until your arms break off. Now, that being said, if this is the phospholipid bilayer right there, what are the amino acids up here? How would you describe them?
Alpha and beta.
No. Scale it back a bit. Okay. Phospholipid bilayer. All right? If that's the bilayer, how would you just chemically describe those amino acids? Because what's up here? If this is the bilayer, the heads are facing that way. Water, so these are termed what? Hydrophilic. And then right in here, what are they? Hydrophobic and then hydrophilic. They've got the same motif. All right, so you've got this plasma membrane made up of a phospholipid bilayer completely surrounding the cell. Next up, we have a cell wall. Some plants have them, some plants don't. Some cells have them, some don't. Plants definitely have them. Certain fungus have them, but not all. The cell wall is usually made up of a very, very rigid carbohydrate. What kind of structure would that rigid carbohydrate have if it provides shape to the cell? Branched, unbranched, or moderately branched? It's unbranched because they stack. If it's branched, that's for energy storage. Unbranched is for structure. So you're going to have a lot of unbranched structures in that membrane. I'm sorry, in that wall. It stops plant cells from bursting in pure water.
Oh, yeah. That would be bad for them.
Outside of the cell, you have what's termed an extracellular matrix. Animals have this. It's a whole bunch of things like collagen. They can be connected with integrins, those proteins that we just talked about. It acts as a border between cells. There's a lot that can happen there. You can have junctions allowing for direct transport between cells. It can allow for communication between cells.
Like the slums outside
It's where all the hobos hang out. That's a really tricky way of putting it. Or you can look at it as instead the suburbs, where all the yuppies hang out. A lot of communication going on there. This is between the two. Now, collagen is, you've probably heard of people getting collagen injections, right? So collagen is going to be used for holding your cells together, to waterproof you. Perhaps, right, well, perhaps you've noticed that you don't leak all the time. Like, there's not bits and pieces, not just weeping blood in different parts. That's because you've got this collagen that's really holding your cells together, waterproofing you. I mean, sometimes they work, sometimes they don't. Obviously, if you guys have taken you've taken a shower before. If you took a long shower, you came out and your fingers are all wrinkly, right? A lot of people think they're wrinkly because you lost water. It's just the opposite. Your body's pulling in water. It's sucking in water, and it's pushing against the bonds of the collagen. And that's why you get those wrinkles. So in that extracellular matrix, you have these junctions. There are things that will allow cells to send signals directly between each other. Like a train junction, where you can get on and off a train. And there are a few different types of cellular junctions. Plasmodesmata are in plants. These allow for direct connection between cells through the cell wall. This is going to happen a lot. It'll let water and solutes pass directly. So you have pretty much a tunnel connecting them. As you can imagine, in plants this works out because they need to shift material, a lot of material, through different types of cells. Animals have a bunch of different ones. Animals have tight junctions. These make it like a waterproof seal. The tight junctions are, I tend to think of tight junctions as rivets, if you know what a rivet is. You're to come in, you can pound something in, and it holds these two really tight. So you've got a sort of a junction, and then between the two cells, it extends into the cells, holding them together. It doesn't really allow for transport. It just holds them. Gap? Oh, yes?
Rivet, I was thinking like a hollow screw that you can't unscrew.
Okay, well, I think rivet. Well, right. Oh, that's a good idea. Thanks. Gap junctions. These are sort of like the plasmodesmata in plants. They are open and they let water and solutes flow freely. And then desmosomes. Desmosomes are the strongest of the rivets. They are groups of intermediate filaments lashing these cells together. All right, so we've got the outside of the cell. Now we'll talk about the inside of the cell. Inside of the cell, you have the cytoplasm. That is an aqueous environment that takes up about 20 to 50% of the cell volume. If it's aqueous or aqueous, what does that mean?
Water.
It's got a lot of water. So you've got the cytoplasm, and then you've got the cytosol is the area that fills up the cytoplasm is the liquid that fills the cytosol and the organelles. So you'll notice the cytoplasm fills up everything. The cytosol is that region. Cytoplasm fills the cytosol. So you can think of cytoplasm as the gooey stuff.
Wait, so is that supposed to say cytoplasm is the liquid that fills the cytosol?
Now, in the cytosol, in this area, filled with the aqueous solution, the cytoplasm, that's where you're going to get the buildup or breakdown of macromolecules. You're going to build them up, you're going to tear them down. If you build them up, does it require or release energy? And if you break them down, it releases it. So, Sarah, what kind of reaction is going to build macromolecules? It is, yes. Condensation reaction. That's what builds them up. That's called anabolism. What breaks them down? Do you know that one? Hydrolysis. Good. So catabolism is where you break things down. You get lots of hydrolysis reactions occurring. That's going to release stored energy. The flip side is anabolism. You build things up. Think anabolic steroids. They build muscles. Anabolism builds things and it requires energy. Your condensation reactions. Check this out. So if you take all of the catabolic reactions in the cell and all of the anabolic reactions in the cell, you create what's called the cell's metabolism. Metabolism is just anabolism plus catabolism. So there you go. Okay? So you're going to build things using anabolism, and that requires energy, like dehydration reactions. You're going to break them down using hydrolysis to release energy. And all of that, most of that happens in the cytosol. Now, a lot of people have the mistaken impression that a cell is just a big sack of, like, jelly. And it's not. It's got a skeleton just like you do. It's got to maintain its rigidity. It's got to maintain its shape. Now, you've got different things called actin filaments, intermediate filaments, and microtubules. These build the structures. These keep everything in place, and they don't just sort of float randomly. Everything has a purpose when it's moving. You'll notice here that these organelles are locked in place by the skeleton. The first component of the cytoskeleton are the microtubules. These are long, hollow tubes made out of protein. And they span through the whole cell. They go all over the place. They're highways. I tend to look at them as rail systems. These are what other proteins can move along. They're not necessarily used for structure. They're used for transport, moving things. They're going to grow out of an organelle called the centrosome in animals. A centrosome is made up of a pair of centrioles, if you've heard of them. And all they do is they anchor there at one end and grow, 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 grow. I love in plants and bacteria because they don't have centrioles. They have what's called a microtubule organizing region, which is code for, we don't know why this happens, but it does. It just starts growing from a spot.
Doesn't it pass through parts of the cell wall and link that way?
Yes, yeah, the intermediate filaments will. Not microtubules, intermediate filaments linked to the wall. Intermediate filaments are going to grow and attach to the cell membrane, attach to the cell wall, attach to the organelles. The intermediate filaments give it rigidity and structure, strength. They end up, by binding at different points and pushing against each other, giving the cell its shape. You guys remember building, I don't know, maybe even like last weekend, like forts in your house?
Yeah.
Out of sheets and things?
You take the menus at the restaurant.
You make the menus out of the restaurant. But okay, you've done that before, right? Or somebody said they didn't. If not, you have to go home and do that. What did you always put in the center? A chair, right? Why do you put... some people do. You might have been one who's just like, I'm going to stick everything on the edges.
I just tucked it into the edges
of the couch or something.
Keep it taut, yeah.
Right, but you still had something high up.
Yeah.
You always had an attachment point for it. Something that gives it rigidity. Okay, for those of you who didn't do that, have you ever... I'm asking you. At night, in order to hide your cell phone usage from your parents, did you ever hide under your covers?
Yep.
No. When you do that, you make like a tent out of your head. Like, there's you under the covers, and it's got the sheets coming down. You would be the intermediate filament. The chair in the fort would be the intermediate filament. Does that make sense? It's not just the highest point. It's the point of that's giving it structure and strength. Right, the main support. Oh, if you went camping before, if you've gone camping, you put poles in the tent. You don't just throw the tent on the ground and be like, that's where I'm sleeping.
Yeah.
Unless you have those super cool tents. But otherwise, you're like, you've got to put some structure in there, some rigidity.
Yeah.
That's what the intermediate filaments are. Check this out. They're going to attach to the plasma membrane, right? Oh, it's not on this slide. I'm going to keep talking about the other slide. They attach to the plasma membrane. Part of that intermediate filament is stuck in the membrane. So what does that tell you about that end of the filament? If it's embedded in the membrane, if it's in the plasma membrane, it's hydrophobic. And the rest of it is hydrophilic. And then the final piece are called the ACTIN filaments. These are itty-bitty, teeny-tiny. And they grow by adding monomers to their ends. The thing is, these guys, these ACTIN filaments, really only localize to the edge of the cells. They change the shape of the plasma membrane. They cause it to change form. The way I remember this, and I'm not kidding, is actin causes action. Actin causes the cell to move. So there's the three components. Microtubules that are good for transport and movement. Intermediate filaments that are used for rigidity and strength. And actin that are cause actions to occur on the plasma membrane. They cause it to change shape. Walking along the microtubules to help things move, because the microtubules don't do as much moving as these guys do, are motor proteins. They are going to use energy to change their shape and walk along the microtubules. Literally, like this dinosaur, it has little feet, and it's got these arms at the end, and the feet are sort of walking along. You get a phosphate added, and it changes to this shape. The phosphate gets cut off, and you shift it to that shape. And it moves one step at a time, carrying behind it maybe a vesicle or a vacuole like this. Shifting these things throughout the cell. Motor proteins will allow the cell to interact with the exterior world by manipulating the cilia and flagella. Cilia and flagella are used for movement, motion. Cilia are big, long whip-like structures, and they spin really quickly. The way they spin is, you've got a rotor, and these microtubules grab on, they walk, and then they let go. They grab, they walk, they let go, and that gets the whole thing spinning. Literally like a rotor. Cilia work in the same way. Think about it this way. It keeps building on itself. Each time starting the movement is hard. But each time they do that, it gets easier and easier. Have you guys ever been on a boat with a jerk who tried to sort of rock it? If not, the next time you get on a boat, somebody's going to do that to you. They push down on one side, and then they let go. And they push down on the other side, and they let go, and the boat starts to totter back and forth. Every time they do that, the motions get more and more exaggerated. That's what's and then the boat tips over. In this case, that's what's happening with the microtubules. They pull down, and then they let go. And the other side pulls down and lets go, so you get that back and forth motion, allowing the cell to start moving. Now, cilia are much smaller than flagella. Cilia are used used on much larger cells usually. They're like a whole bunch of little teeny tiny motors that get it moving versus one gigantic motor. All right, so that's what holds the cell together. All that. That was all the cytosol and the cytoskeleton. So now we've got the exterior of the cell, and we're going to start going from the inside out. I'm going to talk to you about how we're making proteins, how we're making polypeptides. First off, we need the directions on how to build a polypeptide. They're contained in the nucleus. The nucleus is double membrane bound. That means it's got two distinct bilayers to it, and that's important for later. You've got, sorry, this pen is dying, one bilayer, two bilayer, or one set of phospholipids here, another set of phospholipids there. That protects it really, really well because you have a hydrophilic region, a hydrophobic region, a hydrophilic region, a hydrophobic region, and a hydrophilic region. There is water in between the membranes. Well, an aqueous solution between the membranes, yeah. But yeah. Its job, the nucleus's job is to protect, organize, replicate, and express the genetic material. Which one? Helm's Deep? Yeah. I went with Helm's Deep because it has an outer wall and an inner wall.
And that's the key for the nucleus.
Yep. The nuclear envelope completely surrounds the nucleus. And that makes it hard to get in and out. You need to be able to get through this. The only way through is with a set of proteins called nuclear pores. Now look how complicated these proteins are. You've got this layer on the outside. Tell me about the amino acids in it. Up here. Hydrophilic. Oh, and then we're going through it. And then we hit an aqueous layer. Hydrophilic. And then another hydrophobic layer. And then we're back to hydrophilic. Those charges and those structures in the amino acids make a big difference here. But the only way in and out of the nucleus is through those nuclear pores. They're like a tunnel going through a mountain. Inside the nucleus, you have chromosomes. You've probably heard of chromosomes. You did probably a lot of work with them before. Chromosomes, this blew my mind when I found out, don't just hold the DNA. Chromosomes are DNA. Yeah. Crazy. Just wrapped around some proteins. So you've got these chromosomes that are made of DNA in the nucleus. And then DNA has all of the directions on how to build proteins. Inside the nucleus, you have another small structure called the nucleolus, the little nucleus. The job of the nucleolus is to build ribosomes. What are ribosomes good for? We talked about it just a few minutes ago. What? They help build something. They build the bonds between peptides. They build polypeptides. What kind of reaction is that? It is a condensation reaction. I'm not going to pick on her. She's got this now. You totally have it. I'm good. So the ribosomes help create polypeptides. You got DNA that can be converted to RNA in the nucleus. So we've got our cell membrane, plasma membrane. Inside there's a nucleus. In the nucleus there's a nucleolus. The nucleus is double membrane bound. In there we have our DNA. The DNA can be transcribed to RNA. And you're also building little ribosomes. We need to get the RNA and the ribosome out of the nucleus. How do we get them out of the nucleus? Through what? Through the nuclear pores. So we get the ribosomes out of the nucleus. We get the RNA out of the nucleus, and they're going to go into the cytosol to create polypeptides. So now we're going to get into this whole process of how we build a polypeptide. Polypeptides, it's not random where they're going. They are tagged as they're being built with a chemical label that says, hey, I'm going over here. It's just like when you go to mail a letter, you have to put an address on it. You just don't just like write somebody's name and stick it in the mail. Unless it's Santa, and then it always works.
Always works.
Yeah.
I think you still have to put one
something.
Or something.
So we've got these polypeptides. Now remember, with polypeptides, they are going to undergo a hierarchy of structure. They're going to end up with a three-dimensional shape. During the process we're about to talk about, they are kept in their original primary structure through these things called chaperone proteins. Pretty much it's like you guys can't touch. So they keep them straight until they get to their final destination. The first place they go as they leave the nucleus is the endoplasmic reticulum. The endoplasmic reticulum surrounds the nucleus. That's a weird looking thing. It's this network of tubes. They're flattened. And they're filled with an aqueous solution. There are two distinct parts of it. The rough and the smooth. What do you think the difference is? Well, one's covered in ribosomes. One of them is rough. Yeah. It's got bumps on it. This is the rough endoplasmic reticulum. It's covered in ribosomes. This is what's closest to the nucleus. The ribosomes come out and they bind to it. The RNA comes out and binds to those ribosomes. And when it does that, it starts to push the polypeptide as it's being made into the endoplasmic reticulum. So let's say, oh, I hate this pen.
This is the dynamic environment, isn't it? So like the ribosomes eventually absorb it, and they don't just stay there. They get used, right?
No, they don't. They end up breaking off and going and doing something else later.
Yeah, I mean, so it's a dynamic environment.
Oh yeah, it's not like they just stay there forever. Yeah. So we've got our endoplasmic reticulum. It is membrane bound, which means it's made up of what? Membranes. Thanks. What's the membrane made of? Phospholipids. The RNA binds and starts to thread in the polypeptide. Here's a better image. I'm not even going to draw it. Look, right here on the screen, you got a ribosome binding to the RNA. They bind to the endoplasmic reticulum, pushing in a polypeptide. In the next lecture, we'll go into detail on what happens there, because that's an important step in protein formation. Now, this in the rough endoplasmic reticulum, you're going to get the proteins originally sorted out. They get tagged. Newly made proteins are going to end up, they could be inserted into the membrane. As they're being built, that's the time to get them sort of the hydrophobic, hydrophilic, hydrophobic regions. And you get glycosylation. Great word. Glyco, when you hear glyco, what do you think? Sugar, because glucose. So glycosylation is adding a sugar, adding a carbohydrate.
Is that image artist rendition?
Artist rendition, absolutely. No, I'll show you what they actually look like in a sec. So, all right. You got sorting proteins, inserting newly made proteins into the ER, and performing glycosylation. Glycosylation is adding a carbohydrate, adding a sugar. The smooth endoplasmic reticulum is attached to the rough endoplasmic reticulum. The difference is no ribosomes. Now this is a real picture. It has lots and lots and lots of twists and turns. Now in biology, whenever you see lots of twists and turns, the reason is to add surface area. The more twists and turns you have, the more reactions you can have. So you're going to get things attached here. You're going to get things taken off. Functional groups get added in here. This is also a place where you build phospholipids. This is where the phospholipids get created. So then they're shipped from here out to other parts in these balls called vesicles. So this is our phospholipid. Again, next lecture we'll talk about how they're actually created. But it all happens in the smooth ER. It is. It's totally crazy. From this endoplasmic reticulum, the polypeptide can move to the Golgi apparatus. This is a series of flat discs that are not connected to each other. They are going to interact by moving vesicles from one point to another. So what that means is, if I was to have over here a flat disk, over here a flat disk, I need to get the protein from here to there. It's going to travel. Well, it's in order to move from the endoplasmic reticulum to the Golgi or within the Golgi, you have to create these things. So what will happen is you're going to create what's called a vesicle. This is going to start getting, there's going to be a ring of actin filaments that form there. Actin causes action, and they start to form a ring around one end where that polypeptide is, and it starts squeezing tight. It's like a pimple. More like a belt on a really large person. If you start, and you've seen people wearing clothes that are way too small for what they should be wearing, right? What ends up happening? That's right, bloop. Thanks, bloop. You're spilling over. The stomach distends. It's like you're cutting deeper and deeper. And let's say this is like a Saw movie or something. If you were to take that belt and keep pulling, what would happen? You split in half. So that's what's happening here. It's getting tighter and tighter and tighter until eventually it splits off. So now you've got this thing called a vesicle. It's just a bunch of phospholipids surrounding a protein. Now we've got to move that polypeptide from here to there. It's got to travel. What's it traveling along?
I imagine it's traveling between two.
What is going to act as like a railway or rail system in the cell? Well, no, we're moving from the endoplasmic reticulum. It's part of the cytoskeleton. The little things, the microtubules. Yeah. Oh, sorry. So you got microtubules here, and then we've got to move from the from one side to the other. The microtubule itself is going to do it. What else do you need? That's just the rail. You need the
phospholipids that push it along.
Well, it's not phospholipids that push it along. What pushes it along? Motor proteins. And the motor proteins carry it over here, at which point it fuses and releases the polypeptide. The Golgi apparatus is good for two things. More glycosylation, because we can't get enough of that. Adding more carbohydrates, and also proteolysis. In proteolysis, protein, lysis, break down. You're breaking down a protein. You're cutting it. Now, a lot of people think this doesn't make much sense. We just built this. Why are we breaking it now? And in order to explain it, I draw your attention to cookies. I like cookies, yeah. Everyone's like, now I'm hungry. When you're thinking about cookies, hypothetically thinking about cookies, if you wanted to make a fresh cookie, a single fresh cookie, okay, like 5, because who eats really one? If you were to make 5 fresh cookies, you're still going to make a batch of cookies, right? You're still going to make 12 or 20 or like a 50, whatever, however many you would normally make. Because it is just as easy for you to make 12 as it is to make 5.
Well, the oven's already on.
The oven's already on. You've already got all the equipment. You're taking the time to do it. You're expending energy. So what this is doing effectively is saying, all right, we've got this single peptide that's really, really long. Let's just cut it now. And now we have 2 for the same price. So the Golgi is important for secretion. The endoplasmic reticulum tends to send things around the inside of the cell. The Golgi tends to send it out. It's like long-distance delivery. So you could release things out into the extracellular environment, sending them out, getting rid of them. And then you finish sorting out these proteins, and from the Golgi, or the endoplasmic reticulum, you can go to any of these other organelles in the cell. You might go to what's called a lysosome. Hey, what does the word lyse mean, or the prefix lyse? Lyse, something that's in your hair. It's like, so think, like Lysol, you're thinking? Ly is the strong base. So yeah, what this does is it breaks things down. Hydrolysis, lysis.
Okay.
So you're going to cut, or you're going to break down macromolecules. And the reason you do that is to recycle the pieces. Recycling is important. Because when you recycle components, you don't have to build it from scratch. Inside of the lysosome, oh, this is where some of that chemistry comes into play. You have to have a low pH. Is that acidic or basic? Acidic. These have a very, very low pH to them. That is how the enzymes, the proteins, work best. How is that low pH maintained? How do you keep that stable? What? Using buffers, right? So you have buffers in there. That keeps the pH really low. The lysosome continues functioning. Now, again, this is an acidic environment here inside the lysosome. What would happen to the cell if the lysosome was to break open and rupture? It would kill like everything in the cell, right? Except it wouldn't. Why? Because the cell has buffers that overwhelm the lysosome's pretty small. You've got buffers that'll overwhelm it. So you keep the cell alive. If a lysosome was to burst inside the cell, nothing would happen.
Just like your appendix.
Just like your appendix. Except then you die. You might have vacuoles in the cell. Vacuoles are great for storage, regulating the cell volume, and helping digest things. They're They're like big empty areas. They're like storage boxes. Plant cells have very large vacuoles. Animals have very small vacuoles. They can help regulate cell volume. Here's what's called a contractile vacuole. It fills up with water, and then you use energy, and you push the water out. That stops these cells from bursting open. Like hydration. They help in digestion. A cell will deform the membrane around the edge and wrap around the food particle. What causes the changes in that cell membrane? What filament? What causes that action to occur? So you see this food is being swallowed up, the plasma membrane's wrapping around it. What is causing that action to occur? Actin filaments. Actin causes action. And then a lysosome will bind and digest that food. Well, a lysosome. Several dozen lysosomes will bind and digest the food. Another organelle is called a peroxisome. Peroxisomes break down hydrogen peroxide. We talked about why is hydrogen peroxide bad? Why is hydrogen peroxide bad for a lot of cells? It does. Why does it wreak havoc? What does it turn into? That steals these electrons like crazy. It's going to create oxygen, but what? Okay, hydrogen peroxide. Peroxide, right, is a term. It's bad. It's bad. Good. It's bad for bacteria because it breaks down into what? Smaller pieces. So okay, yeah, it does. That will steal electrons as an unstable ion. Hey, I'll just tell you, it's a free radical. Free radical, stupid on the test thing.
But that's negative ions everywhere, right? And they grab electrons. Where's that positive ions? So hydrogen ions.
We'll get into that later. Okay. I gotta draw that out. Sorry, I have to really picture what you're saying. So you got free radicals that are that can break things down pretty easily. They break down bacteria. Peroxisomes have a protein in them called catalase. And that catalase will turn hydrogen peroxide into water and oxygen, and that's what releases the bubbles. Bacteria that don't have these peroxisomes, they take that hydrogen peroxide and they turn it into a free radical. Yeah. Oxygen. So that peroxisome in your cells, you've got like the antidote to hydrogen peroxide in your body. Your cells take that and they convert it into water and oxygen. And that's what makes bubbles come up when you put hydrogen peroxide on. Now I'm recording this slide again. Specifically so, it now says, I'm not going to say it because it's not on the slide anymore. Mitochondria will provide the cell with energy molecules. These things called adenosine triphosphate, ATP. We're going to talk a lot about ATP in the future. The mitochondria, to be absolutely crystal clear, is the site of aerobic cellular respiration. Can that be a bonus question, or it can be a major question? Yeah, one of them. Site of aerobic cellular respiration, mitochondria. The mitochondria is the site of aerobic cellular respiration. Now, there are going to be there could be a few hundred mitochondria in a cell or a few thousand mitochondria in a cell. There's not just like 1, 2, or 3, despite what pictures tell you. In this image, each little green dot is a mitochondria. I'm sorry, mitochondrion.
No, mitochondrion.
Yeah, sorry. There are lots of mitochondria in a cheek cell. Your cheek cells don't need much energy, but you can still see. Over here, these bright green ones are heart cells. As you can imagine, a heart cell will need lots and lots and lots and lots and lots of energy. So it has a ton of mitochondria in there.
She was looking for all men.
A mitochondrion is a singular. A mitochondrion
Mitochondrion is from like Star Wars.
that's midichlorian. Yeah, it's a mitochondrion. Okay, here's the deal. If you write mitochondria, I'm not going to be like, oh, use the plural form instead of the singular. Use proper grammar. This is all BS. Why are you doing this to me? So mitochondria, all of them, have a double membrane. What other organelle has a double membrane that we talked about? The nucleus. Good. So double membrane. Oh, and look, there are lots of twists and turns in it. Why does it have lots of twists and turns? Right, increases the surface area, so reactions can happen because the place you get all the energy is right along the edges in here. The more edges you have, the more twists and turns, the more energy you get.
It's like the all crust of.
Right. What?
There's a brownie pan that's curved like this.
Oh, okay.
And you get every piece has a piece of crust on it.
So the inner membrane of the mitochondria has those projections. Those projections are called cristae. You don't need to know that. And then this fluid on the inside. It's an aqueous fluid. It's called the mitochondrial matrix. That's where you're making ATP, adenosine triphosphate. And the final one we're going to talk about is the, I know we're there, is the chloroplast. Chloroplasts are the site of photosynthesis. What is photosynthesis?
Sunlight power.
So, sort of, yeah. It's the first step. You're turning sunlight into sugar. And you're going to require a bunch of carbon dioxide to do it. These chloroplasts are also double membrane bound, just like the mitochondria. Get this, mitochondria and chloroplasts are going to both have their own unique DNA inside of them. They don't have, they're not like built by the other parts of the nuclear DNA. They have their own unique genomes. They're independent, and they reproduce independently. Have you guys ever,
They do meiosis, right?
no, they don't. They do mitosis.
Oh,
They,
okay.
have you guys ever covered up a plant with plastic at some point? No, by accident. It's winter. You left a garbage can lid on top. If you pull it off, the plants are still they're sort of like this whitish gray color, yellow.
Albino.
Right. You've seen this, plants that are that color?
Yeah, you go shrimp.
Your plants die that way.
But before they die.
They turn like that, like a grayish color.
Yeah, grayish color. What happens if you're able to save them and you put them in the sunlight?
They stay yellow for a little while and they slowly turn back to, well,
Right, no, you're exactly right. They stay yellow, and then they over time start turning green again. And that's because the chloroplasts aren't being made until they're needed. Okay, how about this? You guys have been in this situation. Have you, at least you understand the concept. If you've gone to the gym before, you've at the very least thought about going to the gym and said, I should go to the gym.
Yeah.
If you've gone to the gym, you'll notice the first time you go, it's kind of hard. It hurts. You run out of energy fairly quickly. But then you never go again. But then, as you do it progressively more and more and more, it gets easier. Because your cells, as you're requiring them to use up their energy, start saying, oh crap, we need more mitochondria. And they start producing them.
it's like when you walk through like 14 miles with a backpack. One day to the next. It's easier.
Last one, guys. And this is a crucial key concept. And you've probably never heard this before. Some of you may have. It's an idea called endosymbiosis. The mitochondria, the chloroplast, and the nucleus can be explained through an idea called endosymbiosis. In this case, one cell in the ancient past somehow developed a nucleus. And we'll get into that in Bio 102 and how that may have happened. It swallowed a brown algae that happened to be really, really good at producing energy. And instead of digesting it, it said, huh, if I feed you sugar, you'll give me energy. Let's keep you around. So that organism was engulfed with phagocytosis. It was swallowed up. That creates 1 cell, 2 cell. As that cell gets swallowed, it ends up inside a double membrane. It survives.
They like adopt it.
Yeah, they adopt it. They feed it. It's just like what you would do to a pet, right? You let it into the house. You give it food. It gives you something. I don't know, comfort. Oxytocin, right. Later on, another algae, a green algae, got swallowed up by a cell. Now this green algae, we have to feed our first algae sugar. This new one makes sugar. How cool is that? So you get them in there. They're working. What? Glucose. Thanks. I love the random shout-outs from the back of the room. It's glucose. I know that one. It is. They make glucose. So now we've got glucose that's being made in the chloroplast, getting transported over to the mitochondrion, and producing energy for the entire cell. So that's endosymbiosis. A larger cell swallows a smaller cell for mutual benefit. Do you have questions?